Okay, we're moving on to the reproductive system. We're going to start with male and then move on to females. So male reproductive system anatomy. So we're going to go again. We have a tendency to begin at one point and work through the system till we get to an end point. And that's what we do with males and females. We're going to start with testes in the males. In females, we start with ovaries, and then we kind of work our way through in a forward direction. So the testes are surrounded by the scrotum. This is where males produce sperm, which is the male gamete. And they also produce hormones such as testosterone. The microanatomy includes seminiferous tubules, where the sperm are actually produced. You have sertoli, or what used to be called nurse cells, that are found in the periphery of your seminiferous tubules, and they secrete what's called testicular fluid, which helps nourish sperm to mature. And then we have what's called Leydig cells, or interstitial cells, that are found between these seminiferous tubules, and they secrete testosterone. Right, the scrotum is the sac outside the abdominal pelvic cavity that contains the testes. It provides a physical barrier, as well as it's able to respond to temperature change and help maintain sperm viability. So here's a picture of the testes. We have the epididymis to the superior aspect of it. You can see the vas deferens and the tube system that ultimately passes by three other glands: a seminal vesicle, a prostate gland, and a bulbourethral gland, or Cowper's gland, and out in the penis. The skin surrounding the testes is the scrotum. All right, so then those sertoli or nurse cells are found in the seminiferous tubules. They're found to the periphery where you have, excuse me, spermatogonia and a lot of the stem cells, so to speak, of sperm. They deliver nutrients to the dividing cells. They are also capable of secreting what's called inhibin, which regulates spermatogenesis by inhibiting the further secretion of FSH and LH. Right, so what does that mean? It means that we are going to look at a negative feedback loop, essentially, of sorts, for releasing follicle stimulating hormone and luteinizing hormone in men at this point. Women have it, secreted also. And when you're producing enough sperm as a consequence of the follicle stimulating hormone, then the Sertoli cells are going to secrete inhibin that feeds back on the hypothalamus, because the hypothalamus controls all the secretions from the anterior pituitary. And inhibin says, okay, stop secreting what's called gonadotropin releasing hormone and stop secreting so much FSH and LH. Okay, so here we have the testes. They are composed of these repeating tubes called the seminiferous tubules. And each tubule, is the repeating structural and functional unit of the testes. So each tube system, is maturing cells from the periphery toward the lumen to be transported up to the epididymis, which is kind of that brown region to the superior aspect. It's kind of continuation, almost the same shape of seminiferous tubules. Spermatids really, will travel up to the epididymis and go through more remodeling of the cells so that it looks like a nice true sperm. We'll look at some pictures as their intermediate forms and shapes, I guess you could say. And then they travel through the vas deferens, but they'll reside up here in the epididymis until they obtain that nice sperm shape, we call it. Right, this is one seminiferous tubule. And like anything else, this should be repeating all over, just like lobules do, just like osteons and compact bone. The most immature cells are found to the periphery. Thus, those are the spermatogonia. And that's where sperm are going to be derived. And as we go through what's called meiosis 1 and meiosis 2, at the end that you end up with one cell with 23 individual chromosomes. and those are the spermatids. And as they go through this divisional process, these two divisional processes, they migrate toward the lumen, the center of this tubule. And then they travel up to the epididymis and reside there for a while longer until they're completely modified. And then they will then pass through the vas deferens. The spermatids are the cells here with the nuclei that are the dark little ovals that are close to that lumen. There are a bunch here up at about 12 o'clock and 1 o'clock into the periphery here. When you see those little cells, the little ovals, like dark oval nuclei, instead of the big round ones that are found more toward the periphery, the outer margins of that tubule. Those are the spermatids. Okay. Oops, I went the wrong one. Right, so the duct system. Right, the duct system transports the sperm from the epididymis, which is your maturation and storage site for sperm, through the vas deferens, which transports sperm to the ejaculatory duct. Right, it kind of courses anterior to the pubic symphysis, then up and around the bladder, back toward the rectum, and then kind of inferiorly where it meets the glands to the posterior aspect and the inferior aspect. So the vas deferens transports sperm to the ejaculatory duct. The ejaculatory duct mixes secretions from the glands with sperm to create semen. The urethra carries both urine and semen. Surrounded by the penis, it has two different types of erectile tissues. So the duct system itself is just going from, the seminiferous tubules to the epididymis, to the vas deferens, the ejaculatory duct, and then your urethra. But there are other glands and structures along the way that you need to be aware of. The glands, the glands along the way are the seminal vesicles, prostate gland, and bulbourethral or Capras gland. The seminal vesicles lie on the posterior side of the bladder. So this is after the vas deferens has coursed anterior to the pubic symphysis, anterior and superior to the bladder, and now toward the backside. So those seminal vesicles produce alkaline fluid to neutralize the vaginal acidity and fructose to nourish sperm. So the gland or the ducts from the seminal vesicles will merge with the vas deferens to form the ejaculatory duct. And it remains the ejaculatory duct for a very small, brief region until it merges mid-prostate with the urethra. And then from there, you're just using the urethra on the way up. So the seminal vesicles is the first kind of gland that the sperm come across or come in contact with. For sperm to become mobile, they need to have ATP to crank, the units within its flagella to produce motion. And to do that, it takes ATP. And to make ATP, you need some kind of substrate and oxygen. Right? So a significant portion of the secretion of the seminal vesicles is fructose, because fructose is now going to be the substrate in the mitochondria to produce ATP so you can have an undulating tail and produce mobility. So first we feed the sperm, bottom line. All right, then that fluid, you form the ejaculatory duct. It travels into the prostate gland to merge with the urethra. The prostate gland secretes a lot of acidic fluid, which activates those sperm. Prostate-specific antigen comes from there. That's where we generally use as a marker or test for prostate cancer. All right, so now that we've fed them, now we need to kind of wake them up and activate them. And that's what the acidic environment will do. Right, then it passes, now that it's in the urethra, it's going to pass by its last very small pair of glands called the bulbourethral or Capras glands, and they secrete a lot of alkaline fluid as well. and mucus. So the mucus and the alkaline fluid help kind of cleanse the urethra, because formerly, there was urine passing through there, and now you want to have semen passing through there. So it's a good idea to maybe cleanse that tube a little bit, but it also reduces the friction. The mucus reduces the friction, of the surface of the urethra, so that it doesn't impede the passage of semen. All right, then we get to the penis. Contains two major types of tissue. You have the corpus spongiosum and the corpus cavernosum. Surrounding the urethra is going to be circumferentially, kind of like smooth muscle around a blood vessel. It's going to go around the periphery. You're going to have smooth muscle there that when it contracts, it's going to open the diameter of the urethra. So it helps maintain a patent urethra. The corpora cavernosa are two pairs, two columns of erectile tissue, excuse me, which contain most of the blood in the penis during penile erection. So the corpus spongiosum functions to increase the diameter of the urethra, and the corpora cavernosa lends rigidity to the penis. All right, so here we're looking at, let's start at the testes and kind of work our way through. Testes are the white ball. The epididymis is that pinkish region to the superior aspect. Then it leaves as the vas deferens all the way around. Here's the bladder. You have the two ureters there. Right. The seminal vesicles are the first gland that it comes in contact with. Right, so that vas deferens will merge with the duct that's leading away from the transporting fluid away from the seminal vesicle, and it forms the ejaculatory duct. So it's a very brief region, and then it merges with the urethra in the middle of the prostate gland. The seminal vesicles primarily secrete alkaline fluid, but fructose in it, right? So an energy source for sperm. The prostate releases more acidic solution to activate and wake up those sperm. And then the bulbourethral glands are to the right and left, just inferior to that. See those little vanilla colored little glands just below or inferior to the prostate. They produce a lot of alkaline fluid and mucus. We have the urethra, the penis. We have the first blue layer of tissue called the corpus spongiosum that surrounds the urethra, and then the two erectile columns of the corpus cavernosum. If you look at that penis in cross section, then you can see a little more easily the paired corpus cavernosum, and then the urethra to the bottom, and the corpus spongiosum that surrounds it. And I think that gives you kind of a better orientation of where the tissues are located. All right, male reproductive physiology includes spermatogenesis, how we form sperm, the process which produces 4 haploid sperm, which means half the chromosomes that it started with, from one stem cell, one diploid cell. Don't worry about the terms haploid and diploid. Just know that you start with one cell, you end up with 4 viable sperm. In females, that does not happen. In females, we start with one stem cell, an oogonia, and at the end of the process, we end up with 1 viable oocyte and 3 what's called polar bodies, essentially parts of plasma membrane and cytoplasm that will just be kind of discarded. Spermatogenesis occurs within the seminiferous tubules in the testes and includes 3 processes: mitosis, which you are familiar with. We have interphase prior to mitosis, because interphase isn't really part of mitosis. You have interphase, and then you have prophase, metaphase, anaphase, telophase, which are parts of mitosis. And so that the mitosis preceded by interphase is how we keep replenishing our stem cells. But when we want to produce our sperm, we're going to go through the process of prophase, metaphase, anaphase, telophase, and divide the cells to produce two daughter cells, and then divide them again without replicating all the DNA, all the chromosomes, so that at the end of this two divisional phase, you end up with cells with half the chromosomes you started with. Okay, so this diagram is pretty good for showing that because they call it the mother cell up on top. And you over to the left-hand side, it's showing you essentially, what occurs in mitosis. Remember, interphase is not part of mitosis. That's going to occur prior to this. But you have prophase, metaphase, anaphase, telophase. So in meiosis 1, all right, you keep replicating, you keep replicating and producing more stem cells because you have to have something, for all the future cells to grow from. But we're going to have meiosis 1 that includes prophase 1, metaphase 1, anaphase 1, and telophase 1. And then you end up with two daughter cells, which are the ones midway in that blue square on the right-hand side. So we have prophase 1, metaphase 1, blah, blah, blah. We end up with two daughter cells. And then we go through another round of meiosis, which is called meiosis 2. So you'd have prophase 2, metaphase 2, anaphase 2, telophase 2. Tells you you're in meiosis 2. And at the end of that, you see down below that, these are the daughter cells of meiosis 2. You end up with 4 cells. You replicated once before this cell started, so that the daughter cells of meiosis 1 had 23 pairs of chromosomes. And then we split them apart without replicating chromosomes, so that each cell now has 23 individual chromosomes. And that way, when it unites with an oocyte that also has 23 individual chromosomes, it restores the 23 pairs that all your other cells have. All right. You can read below as a review a little bit about mitosis and meiosis there. Not going to get into a lot of detail. In females, it becomes a little more complicated, of course, I will just tell you. Okay, male sex hormones. Male sex hormones. We're going to introduce the hormone gonadotropin-releasing hormone. When we were talking about hormones from the hypothalamus that are the secreting, inhibiting hormones that can be put into the hypophysioportal system that travels to the anterior pituitary to stimulate cells to maybe secrete a particular hormone or inhibit and stop secreting that hormone. And I said, we really don't have to know any of those, but the one we will come back to is called gonadotropin releasing hormone, and that's what GnRH means. Gonadotropin releasing hormone. So that is released from the hypothalamus when we have low levels of FSH and LH. That hormone travels through the capillaries to the anterior pituitary to stimulate the release of follicle stimulating hormone and luteinizing hormone. Now we're getting to those last 3 hormones of reproduction that are produced and released from the anterior pituitary. All right, so follicle stimulating hormone in males, right, released from the anterior pituitary, its function is to stimulate maturation of the sex cells. So it stimulates that process of spermatogenesis. It stimulates, the spermatogonia that you start with to become ultimately sperm. Now, I will tell you in truth that testosterone that's produced kind of next door to it, has a role in that as well, but it's primarily follicle stimulating hormone that is going to, and follicle stimulating hormone has a similar function in females. It's going to stimulate that maturation process of oogonia to oocytes. All right, then we have luteinizing hormone, also released from the anterior pituitary, stimulates interstitial cells to produce testosterone. So luteinizing hormone targets the interstitial cells that are clusters of cells found between the seminiferous tubules. So we'll see a picture of that in just a minute. That testosterone that's produced by the interstitial cells stimulates the development of secondary sexual characteristics, develops bone and muscle and growth. Inhibin feeds back essentially as a hormone that we said was actually released and produced by those nurse cells that feeds back on the hypothalamus to inhibit, gonadotropin releasing hormone from stimulating more FSH and LH to be released. So inhibin turns off the system essentially. It inhibits FSH secretion. So spermatogenesis prenatal to puberty, mitosis occurs, cell division producing clones of the original stem cell, which is called the spermatogonia. Spermatogonia have 23 pairs of chromosomes, or 46 chromosomes. They're found to the periphery, the surface, so to speak, the most outer margins of the seminiferous tubules. And they divide mitotically until puberty. Spermatogonia then differentiate to become primary spermatocytes. So when meiosis occurs, meiosis is the cell division, or you have 2 rounds of meiosis, cell division requiring 2 divisions to reduce the number of chromosomes in half, from what's called diploid number, a 2n, to a haploid n. But all we need to know is that we're going to go through 2 rounds of division, meiosis 1 and meiosis 2. In meiosis 1, we start with a primary spermatocyte and we end with secondary spermatocytes. Now we need to let those divide. So then we enter into meiosis 2. We start with 2 secondary spermatocytes and we end up with 4 spermatids altogether. Each will produce 2 daughter cells. Spermiogenesis is when spermatids grow flagella to become spermatozoa. They can travel up to the epididymis where they become sperm. So there are a couple different pictures to kind of show you the same thing. This picture here, the models in lab, these other big cookie sheet model type ones, that have spermatogenesis generally on the left-hand side and oogenesis on the right-hand side. So you can see, okay, which cells are we starting with? Not the primordial cells, but the stem cell versions of males and females. And then, what enters meiosis 1 and what do you end with meiosis 1? And then whatever you end with meiosis 1 is what you start with in meiosis 2, and then what you end with. So you get a better idea. And so you start with the spermatogonia. They go through mitosis over and over again. One of them is going to be selected to then enter into meiosis 1. You start with a primary spermatocyte, you end with a secondary spermatocyte. So we have here in red to the left, meiosis 1, and then it says meiosis 1 completed, all right? Then we begin meiosis 2. We're going to divide those 2 secondary spermatocytes into spermatids. We end up with 4 viable sperm. They become remodeled, become spermatozoa. They go through several phases of shedding some of that cytoplasm and things there until finally it becomes a real sperm-looking sperm. I guess instead of an immature sperm. It's not an activated sperm, but it's a mature sperm. Okay, so the structure of a sperm, it contains a head, it has a nucleus and an acrosome. The acrosome is kind of like a little helmet it wears. It contains digestive enzymes that are going, these enzymes are going to help break through the cells that are protecting and surrounding the oocyte so that the sperm can reach that cell and fertilize. It has a mid-piece region, which contains a lot of mitochondria, and then the actual tail. Right, hormonal regulation, so help the hypothalamus secretes gonadotropin releasing hormone, which then travels to the anterior pituitary. The anterior pituitary gland secretes follicle stimulating hormone, which stimulates spermatogenesis, and luteinizing hormone that targets the interstitial cells to produce testosterone. The interstitial cells that produce the testosterone targets multiple cells, of the body. It feeds to a certain extent to the seminiferous tubules immediately adjacent to it, where spermatogenesis is occurring to help stimulate that process as well. But it targets other tissues, bone and muscle and fat, and develops, secondary sexual characteristics in males. Is responsible for growth spurts and increasing the size and height of an individual, of their skeletal system. All right, so here we have that there's a mechanism, of gonadotropin releasing hormone to the anti-pituitary, FSH, LH. Right? FSH targets the sperm themselves to mature. LH targets the clusters of cells, that are found between seminiferous tubules. Those Leydig cells or interstitial cells produce testosterone. I think that's really good. Okay, then we're moving on to females. We have ovaries in the females, and similar to the males, the testes, believe it or not, they function to be the source of producing the reproductive cells for females, as well as it also produces hormones. Its function is to produce the female gamete in the form of a secondary oocyte. That's what becomes ovulated and produces female hormones such as estrogen and progesterone. Structurally, it does have an outer connective tissue layer that helps support it in its location. There are ovarian ligaments and broad ligaments, different ligaments that really attach to and support the female reproductive system within the abdominal pelvic cavity. When you do have pregnancy and you're carrying your fetus to term, that's a lot of weight and stress. It needs to be held in place during that period of time. So the outer connective tissue to the ovary allows for a surface for these different ligaments and things to attach to. Okay, so next, leading into the fallopian tubes or the uterine tubes is the opening, and that's called the fimbriae. And you sort of have these, I don't know, I guess you could say they kind of look like rose petals almost. These will secrete mucus and undulate and try to capture and sweep in the ovulated secondary oocyte and transport it into the fallopian tube for possible fertilization. Okay, the fimbriae receive, that's what the fimbriae look like. The fimbriae receive the ovulated oocyte by a sweeping motion, secretes mucus and other chemical compounds, which actually can be used to attract sperm to it, like a chemotactic kind of a function. They sweep and they help to capture the oocyte and then funnel that ovulated structure into the fallopian tubes. So now we looked at the duct system or tube system of the female. So we start at the fallopian tubes or the uterine tubes. They receive the ovulated oocyte that the fimbriae have kind of swept in, captured. It's in the fallopian tubes about a quarter of the way down into the fallopian tube toward the uterus is your site of fertilization, or let's put it this way, the optimal site of fertilization. It's also the site of tubal ligation, getting your tubes tied. Right, so not long after that oocyte enters into the fallopian tubes, that is the best place for fertilization to occur, because in the next chapter or so, we're going to look at development there, and you need to have a few days, about 6 to 8, for cell division to occur before this enters into the uterus and is capable of implanting. If it gets fertilized kind of prematurely, then it's not going to be of the right stage to be able to kind of burrow into, the inner lining of the uterus, and it'll just be eliminated from the body. Okay, so from the fallopian tube, we get to the uterus, which is the site of implantation and fetal growth. And then from the inferior aspect there, we have the vagina for the passage of the fetus and for menstrual flow. Keep in mind, it has a rather acidic environment, in the vagina, and that inhibits microbial growth. Honestly, every opening or orifice of your body is rather acidic, and that's to kill microbes and bacteria so they don't, travel deeper into your body. All right, so here we have a lateral view. We have the ovaries. We have the fimbriae. They look like they touch, but they actually don't touch the uterus. Perhaps I think I read someplace that maybe 1 to 2 fimbriae may actually make contact with the ovary, but otherwise they don't touch. So we have the fallopian or uterine tubes to the uterus, multiple layers of tissue there, parametrium, myometrium, endometrium. We have the entrance exit point, as you could say, the cervix at the inferior aspect, which then leads to the vagina. And then we have external anatomy. And then looking posterior to anterior, anterior to posterior, doesn't really matter. We're just looking at it from a different point of view. In the ovaries, this is where we have oogenesis occurring. The fimbriae upon ovulation will capture that cell with some other cells called the cumulus oophorus or the corona radiata surrounding it, and it passes into the fallopian tube where it should be fertilized about where the little black line there is here, just to the left of fallopian. Travels into the uterus. We have 3 layers to the uterus. We have the endometrium, myometrium, parametrium, the cervix, and then the vagina. Okay, so the uterus has 3 layers. The parametrium is the outermost layer. Again, another good surface for ligaments, to be able to attach to, but also it allows flexibility. The myometrium is the middle layer. That's your smooth muscle contractile layer that has to contract, during childbirth. And the hormone that targets that layer is called oxytocin. That's the other stored and released hormone from the posterior pituitary. Then we have the endometrium. That is the mucosal layer that's, adjacent or intimate with the lumen of the uterus. We have 2 regions to it, the functional layer and the basal layer. It's also the layer, the functional layer, which it is closest to the surface of the endometrium, that will be lost during menstruation. It's also a layer, it is the surface that you will have implantation when you have fertilization. So the functional layer is the more superficial of the 2, I guess you could say, that's closest to the lumen of the uterus. And then deep to that is the basal layer, where we have all these stem cells that replace the functional layer after menstruation, essentially. Remember when we looked at skin and we looked at the epidermis, and we said at the base where the epidermis meets the dermis is the stratum basale. That the epidermis is composed of different stratum, like the stratum corneum and things, but the stratum basale was found on the most inferior aspect. And that's where these cells go through a high rate of mitotic division to replace the cells and push, all the stratified squamous upward, so that as, these cells exfoliate from the surface of your body, those squamous cells, you're constantly replenishing it from the bottom, pushing up. Well, that's sort of what happens here. After menstruation, you need to replace those cells that are on the surface. And that is generated from the basal layer. It's also, when you do have implantation and pregnancy, it provides nutritional support for the embryo until the placenta really develops. The cervix is just the outlet to the vagina. So now we're looking at the uterus, the layers, endometrium, myometrium, parametrium, and the cervix. Oops, I'm going the wrong way, sorry. There we go. All right, the external genitalia. The mons pubis is a cluster or region of adipose tissue, fat tissue, anterior to the pubic symphysis. So it's a fatty, rounded area covered generally with pubic hair overlying the pubic symphysis. Then we have two folds of tissue, a large one kind of superficially and a smaller one deeper to that layer that surrounds and kind of covers the openings to the urethra and the vagina. Labia majora is a hair-covered fatty folds similar to or homologous to the scrotum. Labia minora are folds of tissue kind of deep to that and more intimate with the urethral and the vaginal openings. The clitoris is found into the anterior aspect to the vaginal opening and contains erectile tissue homologous to the penis. Mammary glands, are present in both sexes, but are only function in females normally. Right? They are considered what's called modified sweat glands, so they're technically part of the skin. They are estrogen dependent for development. Their function, multiple functions, but primarily is milk production after childbirth. So we have mammary glands that will produce breast milk. And that's controlled or stimulated by prolactin, one of the last hormones from the anterior pituitary. So prolactin, pro means yay, to increase something. Lactin, is lactose, making breast milk. Transporting breast milk from the glands to the surface of the body are ducts, mammary ducts. When you want to kind of have like what's called download of breast milk, from the glands through the duct, to the surface of the body, that will be stimulated by oxytocin. So again, oxytocin comes back again. That's that second hormone stored and released from the posterior pituitary. So when you're ready to go through labor, oxytocin targets the myometrium of the uterus to contract and expel the uterus. At the same time, that oxytocin targets, the mammary glands and the ducts to pass breast milk to the surface of the body, to the nipples. All right, so here we see breast tissue, we see these mammary glands, the ducts. Glands are the target of prolactin to produce milk. The ducts pretty much are the targets of oxytocin to download, essentially, or transport milk to the surface. Okay, the physiology of reproduction in the female. So in males, we looked at spermatogenesis. In females, we look at oogenesis. This is far more complicated, I'm just going to tell you. So I think once you have lab and you see maybe some of the models, it makes a little bit more sense. But like most things, women are far more complicated than men, especially when it comes to reproductive capabilities and the hormones. So just like men, women are going to start with, a stem cell called, the oogonia, right? Oogenesis, like spermatogenesis, is the process which you're going to make a viable oocyte that's carrying, your genetic contribution, your chromosomes, and 3 polar bodies. So in males, you produce 4 viable sperm. In females, you have 1 viable oocyte capable of fertilization, and 3 polar bodies, which are very, very small, plasma surrounded, and full of a lot of cytoplasm, but not enough organelles to maintain viable life. It occurs in the ovaries, and it includes the same 3 processes: mitosis, meiosis 1, and meiosis 2. Now, there are some major differences with males and females. And first of all, is that females, they're pretty certain, but there might be some new research that might contradict this. I think at USF, they were doing something along these lines, but that when females develop embryologically, they are born with a fixed number of, and not very few, I mean, many, oogonia that will be capable of being ovulated every month during a woman's reproductive lifetime. So you are born with all the cells that will ever be able to be plucked and ripened and send down this, through this process of maturation. So we begin certain phases of meiosis, embryologically, but then it stops, halfway. And then we wait till we start to, during adolescence, to select an egg to mature and ripen to be ovulated. And during that process, then it completes meiosis 1 and enters meiosis 2 and stops again. And meiosis 2 does not become completed unless it becomes fertilized in the fallopian tube. So, like I said, females are way more complicated. All right, so fetal stages, oogonia, developed by mitosis. All oogonia become primary oocytes, and a primary oocyte, or primary follicle, essentially, is one oocyte in the center, surrounded by a layer of either what's called follicular cells or granulosa cells. But they always have like a protective covering on them. The primary oocyte begins meiosis 1, but stops in meiosis 1 at prophase 1 until puberty. So that's another big difference. Women generally are born with a lifetime supply of primary oocytes that are present at birth. They remain inactive until puberty. So for males, it's kind of first in, first out. You just cranking out lots of them once you reach, adolescence. But in females, they're born with, a quantity, a fixed quantity of cells that will go through the maturation process. Females are born with them at birth, and and that's the theory. Although, as I said, I think USF did some research and they said that there's a possibility that at different further stages in female development that they might actually be making more of those oogonias and have newer formed primary follicles and oocytes. Okay, so once a female reaches puberty, we number their cycle based on day 1 through day 28, which is the average, not not everybody has a menstrual cycle or a cycle for 28 days. But we start with generally calling day 1 the first day of menstruation, and then we're going to look at what's happening in the ovaries and what's happening in the uterus. So day 1, a primordial follicle grows into a primary follicle. A primordial follicle, as stated in here, and perhaps I skipped over that, is a primary oocyte surrounded by one layer of cells. One layer of cells. So that starts to, so on day 1, we're going to start secreting follicle stimulating hormone, from the anterior pituitary. And follicle stimulating hormone targeted the spermatogonia to become sperm in males. Well, follicle stimulating hormone targets the primordial follicle to grow into a primary follicle. So we have this cell, the oocyte, in the center with a layer of cells around it. And now it starts to get bigger. It actually will grow in size. And it's going to grow because follicle stimulating hormone is targeting it. About day 5 to day 13, that primary oocyte now completes that meiosis 1. Remember, it stopped in prophase 1. Completes meiosis 1 and produces, boom, one secondary oocyte, just like primary spermatogonia became secondary spermatogonia. Primary oocyte is going to develop into a secondary oocyte. So at the end of this primary follicle is going to split and produce 2 daughter cells, essentially. One is going to be a nice normal secondary oocyte that contains most of the organelles in the cytoplasm. The polar body is a very small cell containing a nucleus but not having very much cytoplasm or organelles at all. All right, so at the end of during meiosis 1 and meiosis 2 in a female, we have unequal division of the cytoplasm or in the organelles, or unequal cytokinesis, because that's what cytokinesis means. So at the end of meiosis 1, you have one oocyte that takes most of everything and gobbles it all up, and it leaves the other one, the little polar body, with like kind of nothing, except its nucleus and a little cytoplasm, a couple organelles. It'll be very tiny compared to your secondary oocyte, your secondary follicle. Once it reaches this stage, that secondary oocyte generally develops 2, if not ultimately 3 layers of those follicular or granulosa cells. And that's kind of how you know that it's a secondary oocyte. It's not only getting increasingly larger, but the number of layers of cells surrounding the actual oocyte has increased. So now that develops into a secondary follicle. A follicle is the oocyte inside and the cells that surround it. We enter into, in this phase now of meiosis 2, because meiosis 2 starts with a secondary follicle, is that it stops again. And it stops again at metaphase. And now it waits to be fertilized. So we have different intervals where things start and then it stops, and it starts and it stops. And that's what's complicated about females. So meiosis 1 starts while you are still in utero, developing before you're born, and it stops at prophase 1. And then once you start to have puberty and you start to have producing eggs that will be ovulated and thus begin menstruations or menses, is that you will complete meiosis 1, enter in meiosis 2, stop at metaphase. And the only way you're going to complete this is if you have fertilization. And if you do, then you're going to divide and have one oocyte that takes the majority of the cytoplasm on organelles again and produce yet another polar body. The polar bodies, that we ended with in meiosis 1, right, or that one polar body, that will divide and produce 2 more polar bodies. So that at the end of the process, we have 1 viable oocyte and 3 polar bodies. Okay, about day 14 is when we have ovulation. The follicle is at its most mature stage, called a Graafian follicle. It's become very, very large, really like a cyst. Some of the granulosa cells get pushed off to one side, and it develops this fluid-filled cavity called an antrum. And now it's about ready to just rupture. It will merge with the surface of the ovary, break down, the connective tissue, and then kind of spit out that secondary oocyte that takes some cells that surround it with it. So it's at its most mature stage. Graafian follicle ejects the secondary oocyte and the polar body because they haven't completed meiosis 2, and that is called ovulation. That is due to a huge surge of luteinizing hormone. With that oocyte, it takes, the structures of that, I guess you say, or the 2 structures you have to know, or regions that you have to know, of this ovulated structure, is the corona radiata, which is the layer of cells that surround the oocyte to protect and nourish it, prevent desiccation or kind of dehydration, drying up. That it was essentially some of these granulosa or follicular cells that were surrounding it while it was still in the follicle form. It takes some of those cells with it, and they're the same cells. We just now call it a corona radiata. And then you have the zona pellucida, and that's a membrane that's between the corona radiata and the oocyte itself, and it contains different receptors called ZP3 receptors for sperm to attach to. All right, so here it shows you a similar picture as we looked at in males. Here are the meiotic divisions, and the follicular development phases. So I think on the left-hand side, it's just a little easier to see, but we have meiosis one that becomes interrupted while you're still in embryologically forming before you're born. And then we divide into two cells. If you work down to the paler greenish region here, each month, from puberty to menopause, one of those oocytes that was halted in prophase one will complete meiosis one and end with one big cell called the secondary oocyte and a polar body. That's where your N is. So you see where it says about 3/4 of the way down to the left, first polar body. Those are the products. The N and the large secondary oocyte is what you're left with at the end of meiosis one. All right. Then we enter into meiosis two. So we divided once. Now we have to divide again. The polar body divides, and upon fertilization, so it shows a little sperm down there. Upon fertilization, then that secondary oocyte will complete meiosis two and produce another polar body. And now to the right, you have an ovum. Now you have a zygote forming. So males end up with 4 viable sperm. Females, no, only 1 viable oocyte. And if that is then fertilized, then it will end up producing 3 polar bodies. Over to the right-hand side, here's a follicular development in the ovaries. Here we have what's called a primordial follicle. You have a little oocyte in the center, kind of like a sperm, but the female version, and 1 layer of cells that surrounds it. So that's what a follicle is an oocyte with some cells surrounding it as a unit. It's kind of, we won't give another analogy. And then that is, again, it's interrupted in meiosis one, at prophase one. As it gets larger and starts to, it comes ready to complete meiosis one, it becomes a primary follicle. So look how much larger it is than the primordial follicle to the top. It's getting larger and larger. It finishes meiosis one and now becomes a secondary follicle where you have an oocyte. Now we call that oocyte a secondary oocyte. And you see how it has multiple layers of cells surrounding it. And that's how you can recognize a secondary oocyte on a slide. It matures more and more to the mature graphium follicle. You can see this fluid-filled cavity there, pushes some of those cells off to the side. And then upon ovulation, a huge luteinizing hormone surge. That structure is going to merge with the surface of the ovary and release your little secondary oocyte there with its granulosa cells surrounding it. It now becomes called the corona radiata, and it leaves behind the rest of those cells in that fluid-filled area. And that those cells and fluid that it leaves behind is called the corpus luteum. If there's no fertilization, this corpus luteum will degenerate into a corpus albicans. But why it's still viable like this, and in yellow, right, means it's secreting steroid hormones. So luteinizing hormone from the antipituitary is going to target this so that it releases estrogen and progesterone. So it becomes a little more complicated. Menopause. Ovaries lose their function as an endocrine gland, no longer a production source of estrogen and progesterone. With the loss of estrogen, you have a couple things that start to change. One is you have the breakdown of bone, loss of calcium uptake, and loss of inhibition to osteoclast. So it affects some of your calcium uptake from the digestive system. You also lose some of the inhibition to osteoclasts. Remember, osteoclasts break down bone. It can develop into certain cardiovascular issues because we don't have estrogen that helps regulate cholesterol levels, so to speak. And it can lead to the atrophy of reproductive organs and the breast tissue. With lack of estrogen, it also allows smooth muscle. That's how it often affects or develops other cardiovascular issues, is that smooth muscle of the, all smooth muscle locations, but a lot of it along the urinary tract and in the reproductive organs, and some blood vessels will actually start to atrophy. Okay, we're going to talk about menstrual cycles versus uterine cycles and how they overlap. All hormones, FSH, LH, estrogen, and progesterone, are low when we begin this cycle. So whenever we're talking about a cycle, we're talking about, a monthly cycle a female goes through. The ovaries go through a certain process, and then the uterus goes through a process. We all start, or both cycles start on day one, and day one is the first day of a menstrual bleed. So the ovaries are preparing. The ovaries' job is to ripen an oocyte and ovulate it and put it out there to be captured into the fallopian tubes and be fertilized or not. That's its only job. So the ovaries keep grabbing one of those primary follicles, it matures it, it lets it ripen, become a Graafian follicle, spit it out. That's all it does, around and around and around. All right, goes from the primary follicle to secondary follicle, ovulation. The corpus luteum remains behind, producing estrogen and progesterone. We'll cover that. And that's it. If you have fertilization and you have implantation and pregnancy, this system is going to stop. We're going to talk about that in a subsequent chapter. But if you don't have fertilization, then back to day one, it starts maturing into the primary oocyte and getting it ready about day 14 to be ovulated. And that's its only job. Right. So the first about 12 days of an ovarian cycle is called the follicular phase. Why? Because the hormone that's primarily being secreted and working right now is follicle stimulating hormone. We're going to ripen that primary follicle into a secondary follicle and get it into its meiosis II and get it ready to be ovulated. About day either 12 to 14 of the cycle, it's a single event of ovulation. It's going to release that secondary oocyte so the fimbriae can capture it. And that is due to a big surge of luteinizing hormone. After that, we have the remaining about 2 weeks to cover, and that is what's called the luteal phase, from day about 15 to the end of the cycle, about 28. And we call it luteal because the hormone that's primarily being secreted here is luteinizing hormone. I mean, it surged at about day 12 to 14 to stimulate ovulation. It'll drop down some, but that luteinizing hormone is going to target that corpus luteum to keep producing estrogen and progesterone in the event that we have fertilization and implantation. Okay, so that's the cycle. The ovaries go around and around and around and around. The uterus has a different function. All it's meant to do, and it goes kind of around and around and around, is to become engorged with blood in the event that you have implantation that has a ready blood source, to develop a placenta so that we start having an environment where this embedded structure can survive for 9 months and then get expelled at the end of 9 months. So the uterus engorges itself, prepares itself with a high degree of blood vessels and highly vascularized in the event of that there's fertilization and implantation. And if it doesn't happen, it goes, okay, well, we need to peel off the surface and start all over again. And that's what menstruation is an exfoliation of that, functional layer of the endometrium, of that inner layer. So it becomes engorged, and then it needs to essentially, like having a bad sunburn, is peel off the surface and let the skin grow back again. And that's what the uterus does. It goes around and around and around. If you have fertilization and implantation, then that process will stop, of trying to exfoliate, and hopefully because you have estrogen and progesterone. If you do not have implantation, then you will have menstruation. and it will start back over again to then redevelop and regrow and engorge the endometrium for another cycle and possible opportunity for pregnancy. Right, so the three phases of the uterus are the follicular phase, the ovulation, and then the luteal phase. So in the follicular phase, about day 1 to day 12, FSH stimulates follicular growth. Meiosis restarts, the primary oocyte changes to a secondary oocyte because it completes meiosis 1, enters meiosis 2. Luteinizing hormone stimulates the granulosa cells surrounding it to release estrogen. Not a lot of luteinizing hormone, but there's luteinizing hormone. But about day 12 to day 14, we want to release that secondary oocyte. And so we have this huge luteinizing hormone surge, and that stimulates ovulation. Following that, we had the luteal phase about day 15 from ovulation to the end of the cycle, essentially, about day 15 to day 28. Luteinizing hormone stimulates the granulosa cells of the corpus luteum, that portion of that mature follicle that stayed behind, and it secretes progesterone and estrogen, or P and E. Those two hormones in different ways maintains the endometrium of the uterus in the event that you have implantation. If there's no implantation and no pregnancy, that corpus luteum that was left behind is going to degenerate and it becomes, what's called the corpus albicans, and albican refers to white. So it becomes essentially a scar tissue, no longer releasing or producing estrogen and progesterone. All right, so here we can see our ovarian cycle, right, where we have a primary follicle, secondary follicle, mature follicle. Right, in blue, right, that represents the follicular phase. So down below that goes, okay, here's your follicular phase, right, about day 1 to about 12 to 14-ish, right? On the average, 14 is your day of ovulation. All right, so that just represents the blue segments there, follicular phase. Ovulation, a single event. And then, so we're talking about the ovaries. What's left in the ovary? That is the corpus luteum. All those cells in the fluid inside remains as the corpus luteum, releasing estrogen and progesterone. Because we're secreting luteinizing hormone, and that maintains that corpus luteum so that we have estrogen and progesterone. And so essentially, we don't start to menstruate. We give that implanted structure a chance to develop and start creating its own hormones of communication. If there's no fertilization, it degenerates into a corpus albicans, which is over to the right-hand side. And then we resume back to blue. We resume back to day 1, and we go right back to the beginning and go through this system or these phases over and over again. The uterine cycles are different. We have 3 phases: a menstrual phase, a proliferative phase, and a secretory phase. The menstrual phase includes day 1 to day 5. That's what's happening when the endometrium is essentially exfoliating, and allowing it to be shed. It takes about, on the average, 5 days. Then we enter into the proliferative phase. So once that surface, that functional layer of the uterus has exfoliated, so to speak, we need to build it back up again from that basal layer. That takes about another week, from day 6 to day 14. And then for the remaining 2 weeks, about day 15 to 28, we call it the secretory phase. And this is mostly the action of progesterone. The actions here in the proliferative phase is mostly estrogen. So during the secretory phase, we want to now engorge and develop blood vessels extensively toward the surface of the endometrium, so that if something implants there, we will have a ready blood supply to start to develop the mother side of nutrition. All right, menstrual phase, day 1 to day 5. We have low progesterone levels that cause the endometrial lining to shed, and thus we have menses. FSH and LH levels begin to rise because even though you're menstruating, the uterus is going through this phase, the ovaries are already starting to stimulate with FSH, one of those primary follicles, to start maturing. During the proliferative phase, about day 6 to 12, increased LH levels cause increased estrogen levels. So essentially the estrogen levels are going to increase. Estrogen stimulates the repair and rebuilding of the endometrial lining by increasing mitotic rate, increasing mitosis in the endometrium. In the secretory phase, which is about from ovulation to the end, about day 15 to 28, progesterone stimulates the endometrium to prepare for implantation and to grow by increasing the blood supply and the mucus secretions there. Progesterone also stimulates the formation of what's called the cervical plug to block any further sperm entry into the uterus. If there is no fertilization and implantation, progesterone levels fall due to the corpus luteum degenerating, uterine artery spasm cutting off the blood supply to the surface, and thus the surface endometrium dies off. And now it needs to essentially exfoliate. Right, so this shows you essentially what's happening. If you look to the left-hand side, it's indicating the functional layer and the basal layer. We said the functional layer is the surface of the uterus that's exposed to the lumen of the uterus. And it's the functional layer that exfoliates and is replaced during menstruation. So at the beginning, starting with day 1, now on the x-axis, we have the first 5 days, it's the menstrual phase. So when we're looking at the thickness of the functional layer, it's become minimal at the end of 5 days because it's essentially peeled off, right? It's exfoliated. But now we go into the proliferative phase. We have a lot of estrogen increasing the mitotic division from that basal layer in the darker pink upward. And so just like in the skin, that where we have high mitotic division of squamous cells at the stratum basale, right, is pushing all those cells and increasing, or replacing cells in the epidermis. It's doing this, replacing the functional layer of the uterus. So it gets thicker and thicker. That's your proliferative phase. And then look where it turns into the green region from about day 15 to day 28. That's the secretory phase. And now look at these blood vessels become large and engorged and supplying the surface, the most superficial, surface of your functional layer in the event that you have something implanted. If we don't have implantation, then we're not going to continue to secrete luteinizing hormone. We do not have more estrogen and progesterone being secreted from the corpus luteum. And with the drop of those hormones there, about day 25 to 28, to twist off. You see how you get to about day, 26, 27? They start to twist off at the bottom. And once you cut off the blood supply to the surface of the endometrium, the tissue dies off, it needs to exfoliate. And that's the cycle of the uterus. The uterus just prepares for implantation, and if it doesn't happen, it sloughs off and it gets ready again. The ovaries, begins with maturing a primary follicle to become ovulated, waits to see if there's this fertilization implantation. If not, okay, we're going to start this process again. But they coordinate with each other. Hormonal regulation of a cycle. So the hypothalamus produces gonadotropin releasing hormone, GnRH, just like in males at puberty. It then targets the anterior pituitaries to secrete follicle stimulating hormone, which stimulates oogenesis and follicular growth. Luteinizing hormone stimulates estrogen and progesterone released by the granulosa cells. It also, when it surges at day 14, stimulates ovulation. Luteinizing hormone transforms the follicle into a corpus luteum, and it stimulates therefore the continued production of or release of progesterone and estrogen. Okay, so similar to what we looked at in males, we have gonadotropin releasing hormone from the hypothalamus, targeting the anterior pituitary. Anterior pituitary secretes FSH, LH. So you can see how that works there. The questions I generally ask you are fairly basic from this and are generally types of questions and functions of hormones, what they target, where they come from, that are reinforced in lab. Okay, so, this is the basic cycle. If we don't include fertilization, anything with pregnancy, you can see the cycle of the ovaries and the uterus are they're complicated, but they're pretty straightforward. Once you start introducing pregnancy, then it takes on yet another whole level. So it says, if there is fertilization, in the event we have fertilization, that we've ovulated and sperm have reached that oocyte and fertilized it, then we produce what's called a zygote that ultimately becomes an embryo. And once it's implanted, that structure produces the hormone human chorionic gonadotropin, or HCG. And that comes from the embryo itself, from certain cells that are there as part of kind of placenta, or the fetal side of the placenta. And that then targets the human growth hormone, acts like luteinizing hormone of the mom, and it targets her corpus luteum to keep releasing estrogen and progesterone. Because if you drop the estrogen and progesterone, or those levels drop, the female is going to have menstruation, and this embryo is going to be swept away. All right, so it's kind of the way this embryo is going to talk to its own mom and say, you know what, I'm here, I'm embedded in here, I'm secreting this chemical, signal to tell your ovaries and the corpus luteum there to keep secreting that estrogen and progesterone so you don't enter into menstruation. And let me stay here and keep developing. I'll keep secreting this human growth hormone until I'm really fixed in the uterus, and then those levels can drop down, and it's not quite so necessary. But you need to give it a chance to develop, more fully in the uterus, develop a really substantial implantation and placenta, on both the male, the mother, not the male, both the mother's placental side and the fetal placenta. If there is no fertilization, then the corpus luteum degenerates because luteinizing hormone has now dropped significantly. If there's no corpus luteum, there's no estrogen and progesterone. If there's no progesterone, then the blood vessels, those uterine arteries, twist off at the bottom and spasm, and it cuts off the blood supply to the endometrium. The endometrium sheds, and that is menstruation. And the cycle begins again in the uterus. Okay, and that is the end of the reproductive system.